Hello, everyone. We are going to start the next presentation, the presentation of Professor Jan Tytgat. It will be a plenary conference with the presentation named Calcitonin gene-related peptides from Venom to druggable targets involved in multiple forms of pathogenesis. Professor Jan Tytgat is a full professor in toxicology and head of the Laboratory of Toxicology and Food Chemistry at Catholic University of Leuven, KU Leuven, Belgium. And he holds a bachelor and a master degree in pharmaceutical science and continued his career by obtaining a PhD in physiology at the medical school at the KU Leuven. From 1990 until 1992, he stayed at the Harvard Medical School in Boston, United States, for a postdoctoral training. He holds the chair of toxicology with tuition and research in different areas of toxicology and in the faculties of medicine, pharmaceutical, and bioagricultural science. He is also director of the division of biopharmaceutical science of Leuven Research and Development, a bioincubator for his research in drug discovery. Jan Tytgat has published more than 150 peer-reviewed articles and reviews in international article journals and has received several prizes, among which the prize of the Research Council in KU Leuven, the most prestigious prize for younger scientists, and an international prize of Dr. E. de Croix of the Flemish Marine Institute for his research area of the kinidor intoxication and drug discovery, starting from marine systems and organisms. Jan Tytgat is also leading a forensic toxicology laboratory in Belgium at the request of the Minister of Justice. He has been elected president of the European section of the International Society of Toxicology from 2004 until 2011. Thanks so much, Professor.
Thank you, Diogo, for the kind introduction. Good afternoon, everybody, dear colleagues. Let me start by thanking Mariana Castro for the very kind invitation. It's a true honor and pleasure being here once more. I always love to come to Brazil to people know this. Colleagues have become really friends over the years, so it's a big pleasure being here. The topic today is not about scorpion or spider toxins or sodium channels or potassium channels, that some of you may know me more in terms of my published career. It is something new that we started some years ago. The title Diogo already introduced, and this is a collaboration we've started with the lab of our colleague Slavika Tudzarova, and she's an assistant professor at UCLA in the United States. And I will show you, even when we don't have, let's say, in vivo or phase 1 data evidence, I nonetheless will show you that in the early stage of drug discovery, you can already have some dreams, you can say, or hypotheses that really can claim or make the claim that the venoms we all love contain wonderful peptides and toxins serving as leads for druggable targets involved in multiple forms of pathogenesis. And the multiple forms of pathogenesis, I should be very grateful to the previous speakers today that already have introduced to you Alzheimer's disease and also diabetes, because you will also see in my research or presentation of today, I will try to convince you that also here we have some examples that may give proof to the title. So without further ado, let's start. So the calcitonin gene-related peptides, perhaps it's a bit less well known to some of you in the audience. We know them since the late 80s, and here is a paper published in 92, where you can see that it's a family of peptides, and as the title suggests, it has not only multiple actions, but also multiple receptors. And this is a bit peculiar for, let's say, a peptide, because we think about peptides in venoms. Quite often they are very selective and potent, so not, let's say, broad spectrum acting. And here, so the calcitonin gene-related peptide and family, as you can see in the abstract of this paper, it can or its effects include rapid changes in neuronal activity, relaxations of many types of muscles, actions on metabolism, changes in gene expression, and also from the side of the receptors, it has been shown that they can really modulate several types of receptors. So this was something very intriguing for me as a start of this study. What are they? So we, in our human genome, we have two types of calcitonin gene, G-protein related peptides, so CGRP, alpha and beta. And we talk about peptides, 37 amino acid residues in length. They have an amidated C-terminus. And there are only minor differences between the alpha and beta isoform. As you can see, only 3 residues difference. And in the literature, in terms of the function and role, you always see, let's say, the effects of the alpha form. So a lot of the results I will show have been executed with the CGRP peptide, the alpha isoform. And here is also a link I will make to diabetes with a peptide that is known in the literature as human amylin. And I will explain further because, or the reason why we also studied the amylin. But as you can see here in this amino acid sequence comparison, the human amylin is very, very similar, at least in the N-terminus, to the CGRP family of peptides. They have only 2 cysteines, so there is an N-terminal disulfide bridge, which is known to be important for function. That has been described by colleagues. So the CG-related peptides, what is their physiology and what is also the pathophysiology? Well, let me start from the gene. In fact, everything is derived from the calcitonin gene. You can see here the, let's say, full representation, and it's composed of exons, introns, and after transcription, polyadenylation, and RNA splicing, we see on the left a calcitonin mRNA part, and on the right you see a CGRP mRNA part, respectively in red and yellow. And then the translation occurs, and just like we know in many venoms and peptides, we have pro peptides, etc. Here also you have a calcitonin precursor, and on the right you have a CGRP precursor. And then there is classically post-translational processing leading to, on the one hand, calcitonin and the CGRP. If you compare the sequences, calcitonin and CGRP are fairly similar, which is logical because also this yellow sequence, if you translate it, it's calcitonin gene-related peptide, so it's not much different from the calcitonin. But my talk will primarily deal with CGRP. And calcitonin, what is known in physiology, it's in fact a peptide or a hormone that crucially controls calcium homeostasis in our serum and plasma, and as such also is, for instance, relevant among many other functions for the strength of our skeletal. Okay, if you look in the International Union of Basic and Clinical Pharmacology Review classification, what is to remember? And that's important for the, let's say, next slides of my talk. So how do we have to see them? Well, let me see here. We have to start with the blue. The blue G-protein coupled receptor. So we talk about a membrane-bound protein consisting of 7 transmembrane helices that really is binding G-proteins from the intracellular side. And the CLR stands for calcitonin-like receptor. And what other people have shown over the years is that to become functional, it co-assembles with a minimal 1 transmembrane small protein called RAMP1. So if you combine here this blue and purple, you have then indeed a functional, what we call CGRP receptor. And the best ligand known so far is, of course, then the CGRP peptide acting on this receptor. That's one. The other one, the amylin, and that's why I presented you the amylin peptide already, is very similar in terms of, let's say, target. So please, for the rest of the talk, remember that you have now, again, a 7-transmembrane protein, G protein coupled, which we call the calcitonin receptor, not the calcitonin-like receptor, but the true calcitonin receptor. And also this one, you see it here on the left in green, co-assembles with RAMP1, and to the best of our knowledge, is mostly activated by the amylin that you can see here. So these two targets are the main, let's say, receptors that we have been studying and the focus of the presentation of today. Why do we have to co-express? In the expression system I will explain to you with RAMP. Well, this is an older Nature paper from 1998 where colleagues have shown that indeed these single transmembrane accessory subunits like the RAMPs, that they do regulate the transport and ligand specificity of the calcitonin receptor-like receptor. And how did these people do this? So if you see here on the x-axis the concentrations, increasing concentrations of the CGRP peptide, and you have, let's say, an assay where you measure function. So here they measured the CFTR, the cystic fibrosis transmembrane regulator, in terms of the currents evoked. Then simply to remember is, if you only express the CGRP receptor and you compare it with the CGRP plus RAMP1, you see a leftward shift, so it becomes more sensitive to react, and also the maximal efficacy is increased. And on the basis of this paper, people now in the field always co-express RAMP1 with the calcitonin receptor or the calcitonin-like receptor. How do we have to visualize? So thanks to the, let's say, structural work by Russo and Hay, we have now a clear insight that these calcitonin receptors, they have, of course, a huge transmembrane section, where in essence, in dark blue, you see the 7 transmembrane helices. But not to forget, they have also a quite important extracellular domain that you can see here in blue. And then the RAMP that I've introduced to you, this single transmembrane protein, in fact, is composed of more than only this transmembrane segment. It, in fact, also has this larger extracellular domain. So what happens now if the receptor sees a ligand, like the CGRP peptide that you can see here in orange? Well, people know now that it more or less intercalates here in the blue structure of the CGRP receptor, and that's how we have to envisage this action or the mechanism of action. If you browse the literature, again, for instance here on the calcitonin-like receptor together with RAMP, and imagine you have here in yellow the peptide, CGRP peptide, it's activated. Well, quite often the literature focuses on the effect of the G proteins, because they are G protein coupled receptors. And in most cases, let's see. Thank you. So in most cases, if you want to know more about the function of the G proteins, they are trimeric proteins of different types. And once this GPCR receptor is activated, then the trimeric G protein splits into an alpha subunit and a beta gamma subunit. And the most well-known studied pathways are the GS type of proteins, and they always involve, let's say, the modulation of adenylate cyclase, and then of course cyclic AMP production and the pKa pathway. And this finally will then lead to effector channels, like for instance ATP potassium channel with a potassium in or efflux. On the other hand, if the G proteins not of the type S are invoked, but you have then the G proteins of the type Q, they're also trimeric, so you have an alpha and a beta gamma. And if these guys, are relevant, then we talk about the PLC pathway, so the IP3 here on the ryanodine receptor or IP3 receptor, and the release of calcium. And of course this calcium is also important to then start a whole type of reactions. But in the literature it's always about GS or GQ, but there are other, let's say, types of G proteins that are involved, and I will show you some evidence for that. But again, to summarize as an introduction what the relevance is of this CGRP peptide and receptors, think about the cardiovascular regulation. So locally, it has been shown that they can be considered as chemical stimuli, mechanical and thermal. So you have here a nerve ending, it releases the peptide, and as a consequence, for instance, it can increase blood flow, it can also be potentially long-lasting, it can promote wound healing and modulate inflammation. And systemically, here on the right, it can be released in response to endogenous stresses with, of course, an effect on the heart, the aorta, the kidney, and as such, from a clinical point of view, you can expect protection against hypertension, hypertrophy, and inflammation. Oops. Doesn't react anymore. Also, the pointer is not. Okay, now it seems to work. Yeah, thank you. I'm saved by the bell. So, CGRP, and what do we know in pathophysiology? Well, the biggest progress that has been made is, for instance, in terms of migraine. So there are drugs on the market to treat migraine, and the mechanism of action is indeed that knowing that CGRP, for instance, in certain blood vessels in the central nervous system causes vasodilatation, then to use antagonists or even antibodies to counter effect, and this is really beneficial to treat people with migraine. So that's one proven, let's say, target. But not only in the brain, so here you see a typical C-type or A-delta fiber neuron with one end connecting the central nervous system and the other one in the periphery. So here you see again the release from the sensory neuron of the CRG peptide. It can interact with the receptor, with then this small ramp, and this is then the mechanism of action to explain the treatment against migraine. But also here, if you see an arteriole or a smooth muscle vessel, there also the same peptide is known to be released from the sensory neuron with a dual type of action, so it can interact with its receptor and then activate via the adenylate pathway and the pKa to produce this NO pathway, and the NO on the one hand, but also a direct action of the receptor on the vascular smooth muscle, and again lead to the pKa activation and then release of potassium, so the potassium efflux, giving a hyperpolarization of the vascular smooth muscle cell and then regulate the tone of the blood vessels. So you see CGRP peptides, they have almost a hormonal type of action, not only in the central nervous system, but also in the periphery. So it really goes beyond migraine, and an interesting, let's say, observation by these colleagues I read was that CGRP can be considered as a class of family peptides that have a more distant site of action as opposed to fast synaptic transmission by neurotransmitters. So we all know the classical neurotransmitters like dopamine, acetylcholine, etc. They quite often act immediately, fast, but locally, where they are, of course, secreted. Whereas if you consider the CGRP peptides, they are released and they travel around in our body as if they were hormones. And this is interesting because if you have a peptide being released and it's behaving like a hormone, then of course you can explain several druggable targets where then in pathophysiological conditions, let's say some organs cause problems. In the CNS, in the PNS, gastrointestinal, in our immune system, at the level of cardiovascular, pulmonary or reproductive systems. So are there more clear examples? Yes. I showed you already for migraine. Then we talk about antagonists. They are on the market. You also can have examples where you need agonists, like atherosclerosis, heart failure, myocardial infarction, pulmonary hypertension, wound healing, aging, still question mark. Also here on the left, hypertension, sepsis, and arthritis. But what was really for me interesting over the last couple of years is to study more in depth the involvement of these peptides in diabetes, type 2 diabetes, and also obesity. And the aspect of obesity has already been, let's say, brought forward today, for instance, with Ozempic. So, as such, we were interested, and here I come back again to the amylin, because you can wonder what does amylin have to do with the family of CGRP peptides and calcitonin. I don't immediately see the relation, but let me explain. Amylin here is in fact a synonym for what we know, islet amyloid polypeptide sequences, so IAAP. And if you blast the sequence, so in an human, you can see again, you talk about fairly the similar peptides as CGRP, 2 cysteines, so also in the N-terminus you have this 1 disulfide bridge, and they are abundantly expressed in humans, also in rodents like rat and mouse, monkeys, porcine, cow, cat, dog, etc. So they're quite abundantly over the different species present. And interestingly, rodents only, so it has been published by other colleagues, if you see rodents, they have a quite peculiar N-terminus. Here you have 3 prolines, and these 3 prolines, according to these researchers, avoid the oligomerization and avoid plaque formation, which for the study and to understand, for instance, Alzheimer is very crucial. So these 3 prolines, and we don't have these, and many other species also don't have this. So in a certain way, rat and mouse are a bit protected in terms of amyloid beta plaque formation with this type of peptides. But here now comes the hypothesis we wanted to go look for, and also the relation to the venoms. IAPP, or the amylin, can exist as a monomer, but it's known to form oligomers, and then it becomes more toxic. And certainly, when we talk about the amyloid fibrils and plaque formation, people know, yeah, this is very toxic. And if you see from the left to the right, it has been shown that in type 2 diabetes mellitus, T2DM, the pancreatic beta cell dysfunctions and gets in a certain stage of apoptosis once you have these amyloid plaque formations or fibrils, let's say, entering the pancreatic beta cells. Moreover, as the title of my presentation suggests, yeah, in many forms of pathogenesis, so the amylin or the IAAP peptide, it has been shown by people that this is another key molecule not only involved in diabetes, but Alzheimer's disease. And the amylin receptors that I have introduced you in one of the first slides seems here also to be a potential therapeutic target for Alzheimer's disease. And also recently, the strategies for treating Alzheimer's disease may be related to insulin signaling, and so there may be a connection. So all this was literature evidence that really took my attention. I said, well, what can we learn from this? Is this a proven link? And how can venoms and toxins help us? Why? Because also in this, I think, very, yeah, perhaps simple, but nonetheless important representation how the disease may occur, and be linking diabetes here on the left with Alzheimer's disease. So remember that then the amylin shown here in light blue, so the human IAPP or amylin, it's a synonym, is being released from the islets in the pancreas, and it's shown to be able to cross the blood-brain barrier. And then in the central nervous system, this amylin has an, let's say, interest to assemble and in fact almost neutralize the amyloid beta, as you can see here, the light blue and the purple blue. And also at the level of the fibril states, they can really, let's say, start a marriage and go together. Whether this is beneficial or not, that is still a bit up in the air, but they definitely have some interactions, and that is, I think, quite interesting to observe. Now, the amyloid beta, and this also has already been presented this morning, we did not study, so for a good understanding, when you talk about amyloid beta, it is not a CGRP-related peptide. It is not the amylin. So this is a different type of sequence. But the amyloid beta, if you consider the amyloidogenic pathway, is the result of some enzymes like the beta-gamma secretase coming from this precursor protein IAPP. But for our good understanding, again, this is not human IAPP, not the amylin. Okay, now the venoms, because we are in a venomous toxinology, let's say, meeting. In 2019, the Australian group of Brian Fry published all of a sudden, in fact, that the vampire venom, so the bat, the vampire bat, shows some vasodilatory mechanisms in his blood. The animal is called Desmodus rotundus, when he's feeding on prey, and the blood may contain some interesting peptides. So they published here from the vampire bat a peptide that you can see. Again, 37 amino acids residue in length, two cysteines with one disulfide bridge, and they compared it with the rat and the human. Very similar, except C-terminus, 9 amino acid residues difference. So the vampire bat indeed also has a calcitonin gene-related peptide sequence, and they have shown in that paper that it can cause, as expected, a relaxation of the arterioles, so the smooth muscle cells, and that both the vampire bat and then the rat have the same affinity, and you can see it's a very potent peptide, so we talk about really a very, very high affinity. Again, this was very interesting for us because this vampire bat and this CGRP-related peptide, how can it cause then the relaxation? Because in the paper by Brian Fry, this was only discussed. Mechanism of action, not known. So we wanted to clarify this. And going back to earlier papers, this one in Nature in '88, here these authors have for the very first time, at that time, published calcitonin gene-related peptide acting as a novel vasodilator neurotransmitter in the mesenteric resistance vessels of the rat. So we said, what is the mechanism? Because also here, the family of peptides was presented, but again, the mechanism of action was not clear. And again, as you can, when you browse the literature, and you see here in blue the typical calcitonin-like receptor with the RAMP1, always we can see an explanation in terms of on the left, the GQ type protein cascade or the GS type cascade. That's always coming back. But we wondered, what about then the beta-gamma subunits that you can see here in brown? Because they seem to be forgotten, although some people have suggested that they also may be involved in the vasodilation. So to discover this or to elucidate and clarify this, this is the methodology we've been using for many, many years in my lab. So the success, let's say, of this validated and open, robust system is the following. We start, of course, with a high-expression plasmid vector in which we can subclone whatever gene that is known. So in my lab, we have now about more than 70 clones, cDNAs, encoding for a wide variety of voltage-gated ion channels. So all the sodium channels of the human genome, invertebrate sodium channels, bacterial sodium channels, calcium channels, potassium channels. You have ligand-gated channels that we can express. Think about the nicotinic acetylcholine receptor, think about the GABA-A receptor, think about TRP channels, and of course the whole bunch of G protein-coupled receptors. Whatever gene you have, you can try to subclone and express. So as such, we can now express histamine receptors, cannabinoid receptors, opiate receptors, bradykinin receptors. This is all possible. Furthermore, with the publication or the libraries where then the genome of many venomous animals are published, you can simply browse and annotate sequences and order synthetically a gene of interest. And this is interesting. For instance, I give one example. If you have a snake, a Mekruru snake, or a pit fiber cobra, and you want to know, is this resistant to its own 3-finger toxins? Well, simply browse the genome, annotate and see where the sequence is of, for instance, an acetylcholine receptor in that species, and maybe it has a different, let's say, sequence encoding for 1 or 2 residues different. You can then order synthetically the gene, subclone it, and then you're in business in a way that we then linearize this. We make in vitro mRNA transcription, so we create M or cRNAs, and then our host cell is an oocyte of this female Xenopus levis animal, So we do once a week an operation, and you can have, let's say, 100 experiments from one, let's say, operation. You wait a couple of days, and then you see here in green the expressed receptor and ion channels at the level of the membrane. And then the essence of the bioassay is, of course, voltage clamp. So we measure as electrophysiologists and biophysicists. With this system, you measure, in fact, current flowing through the membrane, through these green spots of channels and receptors. And the system is like an operating amplifier system where whatever the ion channels or receptors allow to pass in terms of netto flux and current, the system with these two electrodes compensate for that, and it's a compensatory currents that you measure and record. And so in real time you can measure all the activity. So that's the mechanism. And to give you an idea about these peptides, the CGRP peptides, and the next slides I will show with the results. So again, if you're not so familiar with, so if you go from the left to the right, that's a time scale. If you go vertically, that's a current scale. So you can see we measure fairly sensitive. ND96 means a classical physiological buffer in which we allow, let's say, the oocyte that expressing the receptors to sit. For the measurement, we go to high potassium, HK, and this is only to improve the driving force for the potassium influx in this case, to have a really measurable, nice signal. And then at the end of the experiments, we go back to the physiological 97. So at the first moment when we switch from the media, you see an inward potassium basal current. So that is this moiety. That is the representing, let's say, without activation of the GPCR, this represents the potassium current flowing to this blue G protein coupled potassium channels. They are called GIRK. So we express 1 and 2 isoforms. And then, of course, the most interesting thing in terms of toxinology is the application of a peptide. Here in blue, the CGRP peptide. And then you measure this second portion here that is really important to understand how selective it is and what the affinity is. And then we wash out, or we use an antagonist and go back. So that's a typical experiment. And then it means we have success. If you see this result on the left, it means on the right that you have successfully expressed, for instance, the amylin receptor together with the RAMP1, that you have successfully applied amylin or CGRP peptide. You have then successfully, for instance, the GIO or the G alpha subunit from the IO system, dissociated from the beta gamma. And the beta gamma subunit that is known in the literature can activate directly this potassium channel. So that's the mechanism. Okay, now the results. When we expressed or when for the very first time we were successful in expressing these newly cloned receptors after subcloning, etc. So remember, you have the calcitonin-like receptor together with RAMP1. This is the calcitonin gene receptor protein receptor. Sorry for all the letters in abbreviation. So we let it couple to HIRIC1-2. And then we wondered, how does the human variant compares with the vampire or the bat peptide? And here you see an original trace directly taken from the screen. So we start with ND96. You go to HK, you see the expected increase just driven by the potassium channels. And then we applied 1 micromolar of the human peptide, and you see a further increase in current. And then you do a wash out and you go back. So that's a typical example. Then we did the same for the vampire or the bat peptide. And we were very happy to see, in fact, a similar type of activation that we could also wash out, so it was active. But we needed to do some control experiments, because how certain were we that, let's say, the vampire bat toxin from the venom of the bat was not directly activating these potassium channels alone and has nothing to do with the G protein receptor. So then we wondered, can we show that the CGRP peptide from the human and the bat does not direct activate the potassium channels? So we had to do the control experiments, and here I show you the results for the human CGRP. And as you can see, when you have oocytes only expressing these potassium channels coupled to G proteins, you do have the expected change in current with HK, but there is no effect when you apply the peptide. So that was very reassuring. Second question, what do these RAMP proteins do by themselves? Because we co-inject them, And you never know that the co-injection or the presence of RAMP1 may activate an endogenous protein in the oocytes. You never know. So does CGRP peptide direct activate RAMP1, yes or no? Fortunately, it didn't. Here you see a representative example. So both the Venom CGRP peptide as well as the human, you see absolutely no effect when you have oocytes expressing the RAMP1. So these were the control experiments that we did. Then to prove that we really measure on a potassium current, every electrophysiologist will know potassium currents in all these assays are very barium or barium sensitive. And indeed, this is also shown as a control experiment. So we have here, for instance, you have here the, let's say, the effect with the human CGRP. And as soon as we apply 1 millimolar barium chloride, you see a tremendous reduction in current, meaning that it's not a chloride channel that you have activated via, for instance, the calcium release, the GQ type of pathway, because oocytes are prone to have calcium-activated chloride channels, and that's something we definitely did not want to measure. So if it were a chloride conductance, the presence of the barium chloride here would not change or reduce the current immediately. So this is also proof that you really measure potassium currents through the GRK1 and 2 channels, and the same was proven here for the Venom or the bat vampire CGRP. What about then our human CGRP and then the one from the vampire at different concentrations? Well, we have checked it here for the calcitonin receptor, but also the amylin receptor. And on the left, for the human CGRP, we have an EC50 of 50 nanomolar. And more surprisingly, for the bat or the Venom peptide, it's 9 nanomolars. So very sensitive, let's say, corroborating the studies in vivo. So in vivo, people know what these, especially in humans, what these peptides can do. And also in our, let's say, ex vivo system with the oocytes, we are happy that we can say, look, we have similar affinities, so we don't think we are measuring in a bad way. So here is the essence then of the first results I wanted to show. So remember, you have the calcitonin-like receptor here in orange, or the calcitonin receptor. When you combine it with RAMP1, you have then the calcitonin G protein receptor or the amylin receptor. You can apply the human CGRP or the Venom, the bat peptide, so that's the molecule here. As such, it will dissociate the trimeric, go back. Okay, now we're there. So upon activation of the receptor, we know that the trimeric G protein of the type Thank you. So you have a splitting into beta gamma. They activate directly the potassium channel, and then you have the alpha subunit apart. And upon activation of the potassium channel that we call HIRIC1-2, you see this efflux of potassium in vivo, thanks to the, let's say, activation by the beta gamma subunit. So the mechanism of action of this vampire CGRP peptide here on the left, the mechanism of action is, in our system, explainable via activation of a G protein, trimeric G protein, but of the type IO, not GS, not GQ. You can explain everything via activation of these HIRIC1-2 channels. It is barium sensitive, and now, in terms of the symptomology, it leads to hyperpolarization of arterioles. As such, this hyperpolarization will lead to vasodilation of the arterioles, and we can explain the effects of what people have previously shown in a very simple way. You don't need a very complicated pathway. And with reference to Todo Oliveira's group and also Elena Safavi, they have called these peptides, for instance in the bat Venom, doppelganger toxins. So it means that animals have produced very similar peptides as we have in humans, but sometimes also more potent. And this is exactly the case that we saw here with the vampire. And last, there is no participation needed of voltage-gated potassium channels or ATP-dependent potassium channels to explain the vasodilation, nor is there evidence for GS or GQ type G proteins. And as such, we believe we have been able now to explain the early papers of 1988 in Nature, the mechanism of action, what these people first reported as in terms of the relevance of GPC, of the peptides in this way. Okay, now some other Venom explanations. So again, remember, because I know it's a lot with all these abbreviations, when you talk about the amylin receptor, it's always the calcitonin receptor combined with RAMP1. And then the calcitonin-like receptor, if you combine that with RAM, we call it the CGRP receptor. And now I come more to the clinic, because companies like Novo Nordisk, they are making big money, huge bucks, with oral peptides. In this case, a peptide called amycretin, orally available, and they call it a novel unimolecular co-agonist of both the glucagon-like peptide and the amylin receptors that successfully completed phase 1 trials. And what are we dealing with? Well, the oral amycretin peptide in phase 1 is a peptide that is an agonist of the amylin receptor that we also have shown, but at the same time it combines best of both worlds because it's also an activator of the glucagon-like peptide receptor. This is quite interesting, we think. So here you see the amycretin in terms of Novo Nordisk's revenue. It's not too bad, and probably also next year it's going to be in the same direction. And from the European Association for Study of Diabetes, I was struck by this, let's say, slide that you can online see. If you think about Venom's peptides with a dual function acting on both the amylin receptor that we have shown, but also on the glucagon-like peptide, you can best, or you can combine, again, best, or both worlds, a body weight reduction, reduction in appetite and energy intake, and increased satiety. You can control glucose via the amylin receptor, but also the glucagon-like receptor. You can then also think about improving the insulin sensitivity, decrease the glucagon secretion, and delay, acutely speaking, the gastric emptying. And this is the rationale why now Ozempic, Monjari, etc. are so popular. So what we are now busy, as we speak, is also with the same system that I've shown you with the oocytes and the electrophysiology, expressing the glucagon-like peptide receptor, so this GLP-1. In essence, I believe it should work, because we talk again about the G protein coupled receptor like the amylin receptor. In the literature, it's shown that it couples via this GS-type trimeric G protein, via the amylase cyclase, but we are very much interested whether or not we can also let it combine with the beta-gamma subunits of the G protein, so these guys here, and then let it couple to GRK1 and 2 channels. But this is still ongoing. But here comes then our friend, not the vampire bat, but the Heloderma suspectum, so the Gila monster that also has been introduced today with the exenatide that you know. And the exenatide, like almost now 20 years ago, I don't have to introduce it to you, I think the sequence was very much similar to the native glucagon-like peptide. Also C-terminally amidated, like 37 residues length. And then over the years, people have made this peptide more perfect because the exenatide had a half-life very short, so patients had to inject it twice a day, which was not very comfortable. So over the years, companies have then modified the peptide into liraglutide, dulaglutide, etc., where semaglutide is one of the most, let's say, popular ones, also the orally available. And they have made it, let's say, orally available by introducing almost in the middle of the peptide with this C18 fatty acid, let's say, moiety. Nowadays, we are in the phase of extended release formulation of exenatide. And coming back now to my Alzheimer's story, I also found this Alzheimer and dementia paper published recently, 2024 in September, interesting in the sense that this study shows that there is real-world evidence supporting the potential clinical benefits of semaglutide in mitigating Alzheimer's disease initiation and the development in patients with type 2 diabetes. In other words, if you develop diabetes type 2, you may have a higher risk to develop Alzheimer's disease. And the Alzheimer's specialists I know, at least in Europe or in my country, it strikes me that they never, let's say, make this link, or perhaps they don't find it interesting enough, I don't know. But for me this is kind of interesting, because toxinologists as we are, we know the story of the CGRP from this Desmodus rotundus vampire bat and its action on the amylin receptor, we've shown this. And if we now combine this exenatide modified peptide from the heloderma, we may think about indeed molecules that combine best of both worlds. So with in fact an exenatide moiety, with then a CGRP moiety, and then with this carbon 18 type of modifier that makes these peptides orally available. And then the pharmacology of these next generation molecules will be based on explaining that they are agonists for both the GLP-1 receptor and the amylin receptor. What are we also doing now as we speak is checking DACRAs. I don't know whether you've heard about this. DACRA is an acronym for dual amylin and calcitonin receptor agonists. So they are on the market. They are not available as medication, but we are also testing this in our oocyte system. Why? Because you talk about many druggable targets and pathogenesis. Why? They seem to be active in weight loss, anti-nociception, and bone protection. Think about calcitonin, because the bone protection, then we talk about... So... okay. So this KBP 336 can be bought, so we bought it because it's very difficult to synthesize. And here you see the sequence. Remember, you talk about the peptide where in the N-terminus you have these two cysteine residues with one bridge, and then you have here this in the middle here, this heptadecanoyl moiety that you have also seen with amycretin. And in the C-terminal, you have an analog of the salmon calcitonin with a semaglutide building block. Okay, I will finish now. The TRPV1, it also involves, here a recent paper from this year, all of a sudden, TRPV1, a novel target for the treatment of diabetes and complications. So we were wondering, what is this false, is this true? To remember, the TRP channels are ligand-gated receptors. The V stands for vanilloid. So this channel is like the capsaicin receptor, and it's known to be sensitive not only for capsaicin, but also for nerve growth factors, the pH, heat, ethanol. And the principle is that you have an increased calcium influx, and this is the start, for instance, of a pain reaction. As I told you, our system allows expression of many type of channels. So also within the TRP channel family, we have several cloned genes available, among which the TRPV1. And here you see in the raw data. So we can, in oocytes, express TRPV1. As validation, we activate it with capsaicin twice. And then here in the same cell, we applied the Venom or the BAT CGRP, and we didn't see any effect. You could say yes, by itself, it will not have an effect. It should be perhaps an allosteric mechanism of action. So we also checked it in the presence of capsaicin. So the first time, the second time, the TRPV1 also desensitizes a bit, so that's why you see less current. But still in the presence, in the measuring bath of the capsaicin, we didn't see much effect of the CGRP. So in our hands, we cannot confirm the involvement of these CGRP-related peptides into, let's say, nociception or anti-nociception. But the conclusion is, we believe that indeed, if you talk about G-protein coupled receptors, so G-protein coupled receptors here in dark blue, and you activate them by a ligand, do not forget the beta-gamma subunits, because quite often in the literature, it's all, let's say, papers focus on the relevance of the alpha subunits, which is true, we will not deny it or forget it. So then you think about the GI, GS, GQ, or G13, with the famous adenyl cyclase pathway or the PLC pathway. But if you focus on the beta-gamma subunits, we still believe in combination with activation of these G-protein coupled potassium channels, you can think about a druggable target. And a few examples to show evidence of this, that we really have been able to prove this. So we published 2 years ago in the frame of a PhD that also venom of this Automeris zaruma caterpillar, that it contains nasty molecules, among which small molecules like histamine. So here on the left you see again in control in oocyte and the United States you go to HK, but now the oocyte is expressing H4 type receptors, so no CGRP receptors or whatever. So our human genome has 4 isoforms. We have cloned them all, and we have expressed here the H4 type receptor. As a validation, you can apply histamine, so you see the effect. And then, of course, in terms of toxinology, you can start by screening venoms, here the Automeris aruma, and indeed we saw also a huge effect. And later on, we've been able to show that it was truly histamine in the venom that was responsible for this. Here, as a proof also in terms of future drugs, we were able to test this Johnson & Johnson compound, J&J, and it's an antagonist for the histamine 4 receptor. So when you apply here in purple the antagonist, and then you apply histamine and the venom of the caterpillar, nothing happens. So that's nice proof that you really work, pharmacologically speaking, molecularly speaking, on the target. And also for the development of antihistaminics, because there are no H4 antihistaminics on the market, it's only H1 and H2. This also is, let's say, a nice preclinical model to screen and to check. Furthermore, this same venom from the caterpillar was tested on what we call the mass-related G-protein coupled receptors. You will find them in the literature as MGRP-RIX2. Why are they interesting? Because they cause inflammatory pain. So, and here we think about nociception. And they are involved, this type of G-protein receptors, involved in non-IgE mediated pseudo-allergic reactions, which is also interesting. And here, so this is a classical ligand to validate the model. And then we checked the venom, and indeed the venom also has a huge, huge effect. You see the increase in current here when we applied the whole venom on this receptor. And at this stage, for this type of experiment, we still have to elucidate what type of peptide or toxin is involved or responsible for that. So we didn't get there yet. But also the cannabinoid receptors, I'm proud to say that also with another PhD student, we published earlier this year, Stephanoconius species, an oleoyl serotonin molecule that is present in this conus venom. And then with the same type of methodology, we are able to express the cloned CB1 on the left and CB2 receptors. You can apply classical agonists like Win. Win is a well-known synthetically molecule that is an activator. And then you apply, let's say, the conus peptides or screening the venom, and you can also do drug discovery on this field. And then the last one is together with Maria Elena, but I think she just left. So we published in 2018 with one of her PhD students also a synthetic peptide derived from the venom of the Phoneutria nigriventer. It's called PNPP19, and we could show that indeed it's an agonist for particular mu-type opioid receptors. So not the kappa and delta, that's another example, but I show it nonetheless to see or to let you show that indeed it can be used also in the field of pain and opioid receptors, and in terms of venom research, that this model is really validated and an open and robust system. So, and this was published, I see Maria Elena just arriving, so this is the example of the PNPP19 that we have been able to show. And with this, I thank you for your attention, and of course, if there are questions, I remain available. Thank you.
Professor Yuan, thanks so much for your amazing talk. Does we have some questions? Any question?
Thanks, Eve, for a fantastic talk as always. I'm interested on the basics. What would be the interest of the bat to have such a molecule in its saliva? Do you think would be associated with the blood feeding behavior? Will help him to drink the blood easier or release the blood?
Good question, and I can only speculate, but knowing the wealth of, let's say, functions that we know of these peptides in our human physiology, I can only imagine that it must have benefits for the bat, of course. Is it in the level of digestion, level of controlling the blood pressure of the prey or of the bat itself? I cannot give a clear answer, but there must be definitely a benefit that is clear.
All right, thank you.
Someone more?
Excellent talk. I'm still amazed at the results. You mentioned at the beginning that it also the peptide has influence on the inflammatory system, and it acts as a pro-inflammatory, right?
Good question. We don't know again, and I'm happy that you asked the question because maybe I did not stress that enough. A key interest for me now at this stage is to see if there are differences in terms of molecular pharmacology at the level of the monomer, like amylin as a monomer, an oligomer, or fibril, or plaque state. And I hope, let's say, by next conference, with this molecular pharmacology approach, to show differences, because then it can explain, also in the development of pathogenesis of type 2 diabetes and Alzheimer's, then it can, let's say, at the level of basic mechanism, explain what is the relevance of having these peptides in a monomeric state. Are they really pro-inflammatory or pro-predicting a particular disease, a neurodegenerative disease that is developing? Or is the pharmacology of the monomer, the oligomer, or the plaque formation exactly the same? That is something, a crucial element I want to have an answer for, and maybe for the next meeting I will be able to tell you that.
Thank you.
I have a question not related to toxins. I was very intrigued by that strategy, that molecule with there was dual peptides with a C18 you mentioned. Would you know how the C18 works to cross the BBB?
How it works, I don't know again, but it must cross. I think the success of these type of medications that are now orally available and make big bucks is really thanks to this introduction in the middle of the peptide of the moiety that makes it, let's say, resistant against all type of proteases. But I assume it also will not be a hindrance to cross the blood-brain barrier.
That's fantastic.
Yeah.
And do you think would be available, this would be something that we could be applying for our Venom peptides?
Well,
do you envisage that?
That's the goal, of course.
Okay, yeah.
We have the two beautiful animals that help us, the bat and then of course the Gila monster. We could try to mimic it, but on the other hand you can say what's still the novelty. Big companies like Novo Nordisk have been already there.
Yes.
So I anticipate if we really focus on the sequences that we know from venomous animals, we may get the same result, but the novelty is, or we have been scooped already, you see what I mean?
I understand.
But still there is, ample, let's say, possibilities to reduce the peptides and see perhaps where the venomous origin is even a better candidate as the big companies now produce. So that is still up in the air again.
Yes, thank you.
Any more questions? So, Dr. Jan, thank you so much for your presentation.
With pleasure. Thank you.
So now we are closing the oral presentations, and now we can